Hello students and welcome to today's lecture. Today we will be dealing with a very important topic called as the DNA. This DNA is actually the genetic material in most of the multicellular and complex organisms. The word DNA stands for deoxyribonucleic acid. Actually, what happens, this DNA was firstly isolated from the pus cells in 1968 by FREDRICH MEISCHER. FREDRICH MEISCHER at that time had no idea that what he was isolating was actually the genetic material. So he named the genetic material or the DNA as nuclein. This DNA is actually an acidic substance. Now this DNA is further classified on the basis of its appearance or where it is found. So actually in eukaryotes the DNA is mostly found in the nucleus, and such a type of DNA which is found in the nucleus is called as the nuclear DNA. However, in certain autonomous organelles, semi-autonomous organelles like mitochondria and plastids, they also have their own DNA. And such a type of DNA which is found in semi-autonomous organelles are called as the organelle DNA. Now, the DNA which is found in the nucleus has histone proteins bound to it, while the DNA which is found in the organelles, they have no histone proteins attached to it. So this type of DNA which has no histone proteins attached to it is also called as the naked DNA. In prokaryotes, what happens? A proper nucleus is absent. So the chromatin fibers are dispersed in the cytoplasm, and this chromatin fiber further condenses to form the chromosomes. So these chromosomes are found associated with the cytoplasm. So such a type of DNA which is found in the chromosome or associated with the cytoplasm is called as the chromosomal DNA. In some prokaryotes, what happens? Some extracellular or extranuclear DNA or small segments of DNA are found, and this extranuclear DNA is also called as the plasmid. Helically, the DNA can be defined as the double-stranded, helically twisted, long chain of deoxyribonucleotides. Now, what are deoxyribonucleotides? Actually, the DNA is composed of three things. The firstly is the sugar. The sugars, these are further of two types. The first is the ribose sugar and the other is deoxyribose sugar. Actually, the sugar which is found in the DNA is actually the deoxyribose sugar. Second is the phosphate group, which is H3PO4. I told you that the DNA is an acidic substance. Why it is acidic? Because of the presence of these phosphate groups which themselves are acidic in nature. And thirdly, the bases, namely adenine and guanine, which are also called as the purines, while thymine and cytosine, which are called as the pyrimidines. Okay, so these together, sugar, phosphate and the bases, they are together called as the nucleotides. So DNA can be defined as a long chain of poly means many, and deoxyribo due to the sugar, and nucleotides is sugar, phosphate and bases. So DNA can be defined as a long chain of deoxyribonucleotides. Now, how had we determined the structure of the DNA? Actually, it became possible due to two parallel investigations which were going on. The first one was the CHARGAFF'S rule. Actually, the CHARGAFF in 1950 stated that the number of purines is equal to the number of pyrimidines in a DNA, which means A plus G is always equal to T plus C, and this is called as the CHARGAFF'S rule, which helped very much in determining how the bases are arranged in the DNA. Secondly, this A plus T to G plus C ratio is always constant for a species. So furthermore, it helped in determining the species. Second development or the investigation which was going on was X-ray crystallography, which were conducted by the Wilkins and Franklin. Now, the data so obtained from the X-ray crystallography were further given to the James Watson and Francis Crick, which modulated the James Watson and Francis Crick model of the DNA. James Watson and Francis Crick finally gave a model according to which the DNA consists of two strands which are complementary to each other and as well as they are anti-parallel to each other. These two strands are helically twisted in a right-handed fashion, and due to this coiling of the two strands of the DNA, grooves are formed. The first one is the major groove, the length of which is 22 angstrom. Other is the minor groove, the length of which is 12 angstrom. So the one turn of DNA measures about 34 angstrom, and this one turn of DNA consists about 10 nucleotides in it, which means the average distance between two nucleotides becomes 3.4 angstrom. The diameter of the DNA, it is about 20 angstrom. When we talk about the length of the DNA, it ranges or differs from organism to organism. For example, in bacteriophage lambda, it is 48,502 base pairs, while in Escherichia coli, it is 4.6 into 10 to the power 6 BP. BP means base pairs. Okay. Human haploid genome, which means 23 chromosomes, they have 3.165 into 10 to the power 9 base pairs. Now we will talk about how actually these sugar phosphate are bound with each other. Okay. Now before that, these two strands of the DNA, they have anti-parallel polarity, which means the one strand, it is running in 5' to 3' direction, while the other strand is running in 3' to 5' direction. Okay. Now this phosphate group in the DNA is bound with the 2-deoxyribose sugar with the help of a bond, and this bond is called as the phosphodiester bond. Okay. These sugar and the phosphate, they are forming the backbone of the DNA, while this base pair and the deoxyribose sugar are attached to each other with the help of glycosidic bonds. These are called as the glycosidic bonds. Okay. Glycosidic bonds. While the base pairs, they are connected to each other, joined to each other in the strands of the DNA with the help of hydrogen bonds. This adenine is always bound to thymine, and this thymine is always bound to adenine. This guanine always forms bond with the cytosine, and this cytosine always forms bond with guanine. Actually, the pairing of base pairs in a DNA is always complementary, which means they are not binding with the same base pair, whereas they are binding with the complementary one. For example, adenine is complementary to thymine, thymine is complementary to adenine, and this guanine is complementary to cytosine, and this cytosine is complementary to guanine. Okay. Furthermore, this adenine forms double hydrogen bonds with thymine, and this thymine also forms the double hydrogen bonds with the adenine, whereas the guanine, it forms triple hydrogen bonds with the cytosine, and the cytosine also forms a triple hydrogen bond with guanine. Actually, this type, there are 4 major deoxyribonucleotides present in the DNA. What are those? The first one is DAMP. What is DAMP? Deoxyadenosine monophosphate. Second is DGMP. What is DGMP? Deoxyguanosine monophosphate. Third is DCMP, deoxycytidine monophosphate, and the last is DTMP, that is deoxythymine monophosphate. Okay. Now, so this was all about the structure of the DNA. DNA is further classified on the basis whether it is left-handed or right-handed, and also on the number of base pairs which are present in the DNA. So on the basis of it, the DNA has been categorized into these 6 types. Okay, so first is the bDNA, and this was the DNA which was proposed by James Watson and Francis Crick. It is the right-handed type of the DNA, and it consists of each turn, it consists of 10 base pairs. Okay, while the A form of the DNA is also right-handed, and each turn of the DNA, it consists of 11 base pairs. The next is the cDNA. This type of DNA is also right-handed, and it consists of 9 base pairs per turn. Okay, the dDNA is also right-handed and consists of 8 base pairs per turn. This eDNA is actually a synthetic DNA, and guanine is absent in this. So it consists of only 7.5 base pairs per turn. while the Z DNA is the alone DNA which is left-handed and it has the maximum number of base pairs, which is 12 base pairs. So you can learn it this way: E, D, Z, B, A, Z. Okay, so you can just keep on increasing one by one. I hope this was all about DNA. So I hope you like this video. Share this video, subscribe the channel for more. Stay home, stay safe. Thank you.